The plasma membrane or cell membrane is an ultra-thin, fragile lipoprotein and a living membrane that physically separate the intracellular components from the extracellular environment. Various models have been proposed to explain the structure of plasma membrane. Out of them, the widely accepted model is fluid mosaic model. Hello and welcome to SAM online class. Friends, today we shall discuss about various models of plasma membrane. First model is lipid bilayer model. Lipid bilayer model was proposed by Ebert Gorter and Francois Grandel in 1920. According to lipid bilayer model, the cell membrane is a lipid bilayer. This is a lipid bilayer which consists of two lipid monolayers. This is a lipid monolayer and this is another lipid monolayer. So there are two lipid monolayers in a lipid bilayer. For example, the surface area of the lipid bilayer is x centimeter square. Because there are two monolayers in this bilayer, so the total surface area of the two lipid monolayer will be x centimeter square plus x centimeter square is equal to 2x centimeter square. So the total surface area of the two lipid monolayer is double the surface area of the lipid bilayer. Gorter and Grandel, they extracted lipids from the cell membrane of a known number of red blood cells. They spreaded those lipids as a monolayer. After measuring the surface area of the monolayer, they compared that with the surface area of red blood cells. After comparison, they came to a conclusion that the total surface area of the lipid monolayer is double the surface area of red blood cells. So they concluded that the cell membrane consists of two opposing molecular layers. These are the two opposing molecular layers. They suggested that in such a lipid bilayer, the lipid molecules are arranged in such a manner that they are polar hydrophilic head groups, which are represented as red circles face outward toward the aqueous environment, whereas their hydrophobic tails remain embedded in the hydrophobic interior. These are the hydrophilic head groups and these are the hydrophobic tails. The lipid bilayer model of Gorter and Grandel failed to describe membrane properties like surface tension, permeability and electric resistance. They also had a false assumption that the cell membrane consists of mostly lipid. This is about lipid bilayer model. The second model of plasma membrane is the Dobson-Danieli model. This is also known as Pauci molecular model. This model was proposed by Dobson and James Danieli in 1935. According to Dobson-Danieli model, the cell membrane has a trilaminar structure. In such a trilaminar structure, there is a lipid bilayer which is covered on both sides by layers of globular protein. So there is an outer protein layer and there is an inner layer of globular protein. So the cell membrane is composed of a phospholipid bilayer that is sandwiched between two layers of globular protein. Because of this, the model of Dobson and Danieli is also known as sandwich model. Like the lipid bilayer model, this model also had various shortcomings. First shortcoming is that Dobson and Danieli proposed that all membranes have the same structure, but they could not explain that how different membranes can perform different functions. Another shortcoming is that According to this model, the proteins that are present on the surface of the phospholipid bilayer are amphipathic and largely hydrophobic. So they could not provide an explanation that how, being largely hydrophobic, these proteins can be present on the surface of the phospholipid bilayer and how they can interact directly with the water molecules. The cell membrane model of Dobson and Danieli remained widely popular till 1972. The third model of plasma membrane is unit membrane model. This model was proposed by J. D. Robertson. According to unit membrane model, the cell membrane is composed of a bimolecular lipid leaflet that is sandwiched between two protein layers that are organized into pleated sheet configuration. This is a bimolecular lipid leaflet which is sandwiched between two protein layers. These are the two protein layers which are organized into pleated sheet configuration. Robertson fixed cell membrane with osmium tetroxide. After examining, he found the presence of a trilaminar pattern. In such a trilaminar pattern, there was a central light osmiophobic layer and two outer parallel dark osmiophilic layer. The thickness of the central osmiophobic layer was 25 to 35 angstrom and the thickness of the outer dark osmiophilic layer was 20 to 25 angstrom. So the total thickness was between 65 to 85 angstrom. This total thickness was matched with the thickness that was obtained from chemical studies. The unit membrane concept suggests that this trilaminar structure is seen in all type of cellular membrane, including the membrane of endoplasmic reticulum. On the basis of this underlying unity in the appearance of various cellular membrane, this now famous unit membrane concept was proposed. According to this model, this trilaminar structure is seen in various types of cellular membrane. So this model was named as unit membrane concept. The widely accepted model of cell membrane is fluid mosaic model. Fluid mosaic model was proposed by S. J. Singer and G. L. Nicholson in 1972. According to this model, the cell membrane is a quasi-fluid structure. The fluidity of the cell membrane is due to the presence of a lipid bilayer that also provide elasticity to the cell membrane. According to this model, the cell membrane is considered as a mosaic of lipids, protein, and carbohydrate and also sterol. According to fluid mosaic model, the cell membrane is a two-dimensional fluid in which lipids and proteins are capable of lateral diffusion. Because of this fluidity, they can also spin within the lipid bilayer. According to this model, the proteins that are associated with the cell membrane are divided into two different groups: peripheral membrane protein and integral membrane protein. Peripheral membrane proteins are loosely associated with the surface of the lipid bilayer, so they can be easily removed, whereas integral membrane protein are deeply embedded in the lipid bilayer. This diagram explains the structure of cell membrane according to fluid mosaic model. This model considers the cell membrane as a mosaic of lipid, protein, carbohydrate and sterol. So this is the lipid bilayer. In this lipid bilayer, these are the polar hydrophilic head groups which face outward toward the aqueous environment. And these are the hydrophobic tails. Here and there, some lipid molecules are also attached with carbohydrates. So these are the glycolipids. And these are the protein molecules that are associated with the cell membrane. These protein molecules which remain associated loosely with the surface of the membrane are peripheral membrane proteins. And other proteins which cross the lipid bilayer are called as integral membrane protein. There are some integral membrane protein which are embedded in the part of the lipid bilayer, and other integral membrane protein cross the lipid bilayer from one side to other side. There are many proteins which are also associated with carbohydrates. These proteins are known as glycoproteins. There is also protein channel which are present in the lipid bilayer, and this protein channel facilitate the transport of ions. And the integral membrane proteins are of two different types. One class of integral membrane protein span the lipid bilayer only once. Here this is an integral membrane protein which has this alpha helical segment. And this integral membrane protein span the lipid bilayer only once. Such type of transmembrane protein are called as single span protein. Whereas there are other transmembrane protein which span the lipid bilayer several times. Such type of transmembrane proteins are called as multispan protein. And the transmembrane protein can be divided into two different groups, alpha helix and beta barrel. So friends, this is about the structure of plasma membrane according to fluid mosaic model.